Srivastava from UCSF and Gladstone Institute. Yes.
Great. Thank you very much. I'm pleased to be here for this important workshop. So the fact is that heart disease still remains the #1 cause of death in men, in women, and it is the leading non-infectious cause of death in children. And so this talk and the next talk will be focusing on new approaches to deal with this problem. And those who survive with heart disease now have quite a large number. In the United States alone, there are 6 million people with heart failure, and if you look worldwide, that number is somewhere around 25 million. And currently, there's really no disease-modifying therapeutic approach, and thus there's been tremendous excitement in cell-based approaches to address this condition. So the bottom line is, if you have heart failure for a variety of causes, you're left with heart muscle that's not as functional, typically with loss of muscle. So if you want to address this disease, you have to somehow create muscle. And so this slide, as an introductory slide, illustrates some of the approaches that are currently being taken to create more cardiomyocytes in the heart. And there are approaches involving either transplanting or injecting new cardiomyocytes or cardiac progenitor cells that are multipotent and can turn into multiple cell types directly into the heart. And just recently, there's an exciting report in Nature on in non-human primates demonstrating that not only may one be able to introduce cells into the heart, cardiomyocytes, but iPS-derived cardiomyocytes, but also get them to improve function, although there are issues with arrhythmias and engraftment. So these approaches may have potential, but we really have to address the issue of cardiomyocyte maturity that we can derive from pluripotent stem cells, their survival when introduction, engraftment, and electrical connectivity, which are all issues that still need to be addressed, but I think should be able to over time. There is also an investigation of using somehow getting inductive signals into the heart to stimulate resident cells in some fashion to help regenerate the heart, and you'll hear a bit about that in the next talk. But there are a variety of approaches to introduce cells that may not persist and turn into muscle, but somehow stimulate the existing cells and regenerate the heart. The last two approaches are trying to leverage the cells that are already existing with very directed approaches at either reprogramming endogenous and resident fibroblasts into new myocytes, and I'll speak more about that in a moment, or getting the resident cardiomyocytes that are left to re-enter the cell cycle and divide and regenerate the heart that way. I think there's promising approaches here regulating hippo signaling that have potential, and our laboratory has other approaches to try to get these cells to re-enter the cell cycle. I won't speak much about this, but will focus my comments on, given the time, on one approach, which is the conversion of resident fibroblasts into new myocytes. And so the idea is that after injury, regardless of the reason, one might be left with fewer myocytes, and in this case, it's a myocardial infarction, where all this is scar tissue in the left ventricle on this histologic section. The red is normal muscle. And so this approach leverages the observation that, in fact, in the human adult heart, there's an abundance of cardiac fibroblasts. Possibly up to half of the cells in the human heart are not muscle, but rather are fibroblast cells. And so if one could somehow convert those resident fibroblasts in situ within the organ directly into cardiomyocyte-like cells by cellular reprogramming, then one might be able to induce a regenerative effect. So it turns out that no single factor appears to be sufficient to induce this conversion, but inspired by my colleague Shinya Yamanaka's discovery of the combinatorial approach to iPS reprogramming, we leveraged the vast knowledge that we and others had developed over the last 2 decades in understanding the gene networks that control cardiac cell fate decisions during embryogenesis, and screened for the minimal cocktail that would be able to induce this conversion. And it turns out that as we're now finding for many different cell types, a discrete combination of factors was sufficient to induce this conversion. And in our hands, these three factors, GATA4, TBX5, and MEF2C, were sufficient in vitro to push cells toward a cardiomyocyte-like state. There are other combinations that have been reported, including ones from Victor Zhao's lab, using a microRNA-based approach to induce this conversion. But nevertheless, it appears possible with the right cues to induce this sulfate conversion that, in fact, as we've shown, does not go through a cardiac progenitor or a stem cell stage, but goes directly from one adult somatic cell type to another. Now, in vitro, this process was relatively inefficient, and we only rarely saw full reprogramming to a beating cardiomyocyte. However, our goal was really to do this in vivo in the native environment, and it turns out in vivo, this process is much more complete. We have used mice where we can lineage trace the fibroblast population with a fluorescent marker and ask, are those the cells that convert? And by introducing intramyocardially these three genes with a gene therapy approach, we found that we can, in fact, induce new muscle formation. So unlike in this control section, this is of the left ventricle, now in transverse section, all this is scar. When we introduce these three genes, we still see scar, but now have threads of myocardium here throughout the scar region. And these mice, by MRI, have improved cardiac output. And more importantly, the cardiomyocytes that are derived from this conversion are electrically most similar to adult ventricular cardiomyocytes. They're more mature than what we achieve with stem cell-derived cardiomyocytes, and they electrically couple with one another, which we've been experimentally been able to show. So that was good proof of concept, but as I mentioned earlier, the process is relatively inefficient in vitro, and in even vivo we thought we could do better. So we've undertaken a chemical screen to identify chemical modulators that might be able to improve this process, and we've screened a highly annotated library of about 5,500 molecules. And I'll just show you I won't go, for sake of time, go through the data, but tell you that it turns out that both Wnt inhibitors and TGF-beta signaling inhibitors could independently improve the efficiency in vitro, and most remarkably, together, they had quite a remarkable effect. So in vitro, as I mentioned, we don't actually see any beating cardiomyocytes for about 6 weeks after this conversion, even though the gene expression changes occur very, very rapidly. It turns out with the TGF-beta inhibitor, this process becomes not only more efficient, but is accelerated. So within 3 weeks, we see beating cardiomyocytes. And most remarkably, when we combined TGF-beta and Wnt inhibitors, within 1 week, we were able to see beating cardiomyocytes quite abundantly in the dish, looking something like this cell here, which you may be able to see is moving. And so we've taken this further and asked, could these small molecules in conjunction with the gene therapy approach also improve the efficiency of reprogramming and speed and quality of reprogramming in vivo? And so once again, we've utilized the method I indicated earlier, but this time, in addition to introducing the gene therapy, these genes by gene therapy, we've also provided systemic injection of these two chemical inhibitors for 2 weeks. And these are sections through the heart of representative sections through the heart of an animal at the bottom of at the apex of the heart, and in a control setting, and higher up in the heart where there's less scar. And in comparison, this is what we see with the genes plus the chemical inhibitors. And even at the apex of the heart, we see all this muscle and really thick tissue. And you can't see here, but if we lineage trace this with the fluorescent marker, all of this here is fluorescent. The label suggesting that this formerly were non-myocyte cells in the heart. Corresponding to this rapid reprogramming, when we look by MRI at the ejection fraction from these mice, the change in ejection fraction from baseline, in the control setting, either in mice that just had the coronary ligation or had coronary ligation plus chemicals, the inhibitors, and a DS-RED retrovirus, a control retrovirus, you see this decrease in ejection fraction that's typical. Here is what we see with the genes alone, and here is what we see in the decrement in ejection fraction with the genes plus chemicals, which you can see is significantly improved even over what we've reported previously, and that happens as early as 1 week after the introduction of the therapy, consistent with what we've shown in vitro. Now, to translate this towards the human system, we found, disappointingly, that these 3 factors that worked well in mice did not do anything in human cells. And so we had to go back and rescreen for other factors that were missing, and we found that addition of 2 other transcription factors were then sufficient to induce this switch. And so we moved forward with this into pigs, to try to translate this into a larger heart that was more akin to the human heart. And for this experiment, we have induced an infarction by blowing up a balloon in the coronary artery, the left anterior descending artery of a pig at day 0. And then we wait 3 days to let the damage occur and then measure by MRI in each pig the degree of damage so each pig can serve as its own control in assessing the change in its function. And then we've come in at day 5. Here we've used retrovirus for delivery so that we are only infecting the dividing cells. Retroviruses only infect dividing cells, not non-dividing cells. The myocytes don't divide, and the non-myocytes do here in this situation. And then 2 months later, we can look at histology and MRI. So we're still in the process of refining this and have not done it yet with the chemical approach and genes that I mentioned that work so much better in mice. But here, just with genes, what we find is that unlike the control setting where we get a decrease in ejection fraction and then slow further decline over time, we're not only not seeing this decline, but we're seeing some improvement, although not a huge amount, but some improvement in the ejection fraction, and now we're doing it with the chemical approach together with these genes. I should also say that in human cells, with the genes plus chemicals, we then asked, could we reduce the number of genes that are required? And it turns out that we can achieve even better reprogramming with just 3 genes, MEF2C, TBX5, and myocardin, 3 very important developmental genes with the chemicals. And here, within a few weeks, we're getting quite beautiful sarcomeres developing in human cells. And so we're beginning to minimize the number of genes. And so the real challenge now becomes, I think we can do this even in a thicker heart, is delivery. So this speaks to getting at some of the challenges in translation. And it turns out that if we obviously don't want to use a retrovirus that may be integrating in vivo, so an AAV would be a great solution. But AAVs are fantastic if you want to target cardiomyocytes. We have very good AAVs for cardiomyocytes. But if you want to target the fibroblast, they don't get into those, infect those very well at all. And so we have done a positive-negative selection screen in collaboration with David Schaefer's lab at Berkeley to use an error-prone AAV library that can create millions of variants, and then we screen for ones that could infect human cardiac fibroblast but did not infect human ES-derived cardiomyocytes. And we went through 7 or 8 rounds of selection like this, and ultimately have identified several that have greater tropism for human cardiac fibroblast, and one in particular, this one that we've called A2, infects about 65%, with 65% efficiency, human cardiac myocytes, and only about 25% efficiency for the other way around, for fibroblasts with 25% in myocytes. So we have packaged these genes into this viral vector to see if we can get good enough expression, and we're pleased to see that, I can try this video, that with this delivery, okay, I think this is the last, we can get cells beating as early as 1 week, even in human cardiac fibroblast. So I'm going to skip forward to the last slide and just to indicate that as we move forward with this, I think we're beginning to at least believe that this approach can get enough myocytes for improving function, but we really have to tackle the issues of delivery, the regulatory issues around that we can talk about around combinatorial gene therapy and chemical approach are not trivial. And then we still have to deal with safety issues, particularly the potential for arrhythmias. And I'll just say in closing that this approach is not unique to the heart. The same approach is now being applied by many investigators for other cell types for in vivo reprogramming. Thank you very much.